Okay, so let's get started. Good afternoon, everyone, and welcome to day one of the student webinar lecture series by emerging researchers. We are delighted to have all of you join us for this 6-day lecture series designed to create meaningful learning experiences through student-led lectures and expert talks from international institutions such as UCCS and UPR. This webinar series brings together emerging researchers and mentors to explore important themes in life sciences through structured academic sessions. Each day features two interconnected themes, allowing participants to build strong and conceptual understanding while gaining exposure to current scientific developments and global academic perspectives. As part of our Life Science Career and Research program, this lecture series is aimed at helping students strengthen their academic foundations, deepen their scientific understanding, and engage with a broader research community. Today's sessions will focus on cancer biology and cancer therapeutics, covering the mechanisms of cancer progression and the modern advances shaping treatment strategies. To begin today's event, I would like to invite Dr. Sukant Khurana to deliver the opening talk. Dr. Khurana is a scientist, entrepreneur, and founder of the IronCure Group, working at the intersection of AI, neuroscience, and healthcare, with a strong focus on building global research ecosystems and guiding students in their academic and career journeys. Welcome, Dr. Sukant.
IronCure has several young researchers working with us. These young researchers are both teachers and students at the same time. You have to always be a student, but to be a good student, you need to be a teacher. We are privileged to have people work with us who are contributing in an extraordinary way to not just scientific research, to actual product development, to societal outreach, to teaching. These are thorough 360-degree innovators that we have. So we have a series essentially, which is at least in the beginning covering 6 webinar days of 2 interconnected topics. Various young scientists working with our team are teaching in this. These are meant to actually reach to college level, school level, and even young PhD students and provide the perspective which is occasionally or often missing in classrooms. So they're connecting teaching to research and actual impact. The outcomes of these lectures, I hope for people listening, are strengthening of core concepts, but also learning about recent developments and emerging trends. And they encourage scientific curiosity globally and also interdisciplinary thinking. We have, while amazing teachers and students working with us, we would love to tell you what is this as a broader part of things. At IronCure, we integrate 4 things. One, teaching concepts which are missed in college, universities, PhD. Secondly, clear career guidance on PhD, master's, postdoc admission, or careers after medicine. Thirdly, very structured research program with American and European scientists. Fourthly, interdisciplinary thinking, not making you just an academic researcher, but a world leader. People who graduate from our program have gone on to Ivy League, have gone on to top European institutes like EMBL, Helmholtz Institute, various Max Plancks. If you're curious about it, go to AI Vidya or ironcurerx.com or aividya.in or reach out to IronCure team. The details will be in the comments. Okay. I again welcome you to join in this teaching thing, seminars, and understand that learning, listening, understanding, collaboration doesn't end in classrooms. use as many platforms for it, and IonCure provides one platform to bring north, south, east, west researcher, young researcher, bridge those divides. We bring the best from Harvard, we bring the best from Stanford, we bring the best from rural Bengal, rural Kerala. We bring everyone together under one giant virtual umbrella. And I'm so glad to launch this virtual teaching series with our young researchers. Thank you.
Thank you, Dr. Sukant. Now, we will begin the theme one, which is focused on understanding the fundamental biological mechanisms that drive cancer development and progression. So, our first lecture will be delivered by Aditi Dubey, who comes from a biotechnology background. Her lecture is focused on cancer as an evolutionary disease, clonal selection, heterogeneity, and drug resistance. Aditi, welcome.
Good morning, everyone. I am Aditi Dubey, and today I'll be speaking on a topic that has fundamentally transformed our understanding of oncology: cancer as an evolutionary disease. Traditionally, cancer was viewed simply as uncontrolled cell growth caused by mutations. However, modern research has revealed that cancer is much more dynamic. Tumors are not static masses of identical cells. Instead, they are evolving ecosystems composed of genetically diverse populations of cells that continuously adapt to their environment. Much like organisms evolve in nature through Darwinian selection, cancer cells evolve within the human body through mutation, competition, and survival under pressure. Understanding cancer through the evolutionary lens helps explain one of the biggest clinical challenges in oncology: why treatments initially work but later fails. Today, I will discuss how tumors evolve, how heterogeneity develops, and why resistance emerges, and how this knowledge is shaping future therapies. So now let's look into why the traditional view is incomplete. For many years, cancer was described using a linear model. A normal cell accumulates mutations, becomes malignant, forms a tumor, and spreads. While this model is useful, it is incomplete. The modern view sees cancer as a population of millions or billions of cells, each potentially different from others. Within a tumor, multiple subpopulations or clones compete for nutrients, oxygen, space, and survival. Some clones grow faster, some evade the immune system better, but survive therapy. So rather than a single disease entity, cancer is more accurately a constantly changing ecosystem. This shift from a static model to a dynamic evolutionary model is one of the most important advances in cancer biology. To understand this concept, let us connect cancer with Darwinian evolution. In natural evolution, three components are essential: variation, selection, and inheritance. The same occurs in tumors. Variation: cancer cells acquire mutations due to DNA damage, replication errors, or genomic instability. Then comes selection. The tumor environment, immune system, low oxygen conditions, and therapies create pressures that eliminate weaker cells. And then inheritance. Cells that survive, divide, and pass advantageous traits to daughter cells. Over time, fitter clone dominates. This means cancer progression is not random chaos. It is an evolutionary process driven by selection. So now let's look into what is clonal selection. Clonal selection begins when one normal cell acquires key driver mutations that allow uncontrolled growth. That cell divides repeatedly and forms a clone, a population descended from original transformed cells. As the clone expands, additional mutations arise. Some daughter cells gain new advantages, that is, faster growth, resistance to apoptosis, immune escape, and drug resistance. These successful descendants expand further, creating new subclones. Thus, tumor evolves through branching patterns, not straight lines. This model is called clonal evolution, first proposed by Peter Nobel in 1976 and remains foundational today. So, not every mutation contributes to cancer. Tumor genomes often contain thousands of mutations, but only a subset actively drives progression. So, what is driver mutations? These provide survival or growth advantages. Examples: TP53, KRAS, EGFR. Now, passenger mutations. These occur incidentally and do not contribute directly. Understanding the difference is crucial because modern targeted therapies are designed against driver mutations. However, tumors can evolve around these therapies by generating new drivers. Now let's move to heterogeneity. One of the most important consequences of evolution is heterogeneity. This means cells inside the same tumor can differ dramatically. Within one tumor, cells may vary in mutation profile, gene expression, metabolism, growth rate, ability to metastasize, drug sensitivity. This is called intra-tumor heterogeneity. Even two neighboring cells may behave differently. This diversity acts like biological insurance for the tumor. If one treatment kills some cells, others may survive. That is why eliminating all cancer cells is so difficult. As we are talking about heterogeneity, have you ever thought why heterogeneity matters clinically? Let's think about it. Heterogeneity creates major clinical problems. First, biopsies sample only a small part of tumor. That means the sample may miss dangerous resistant clones elsewhere. The treatment response becomes unpredictable. Some cells may die rapidly while others persist. Then heterogeneity increases reoccurrence risk. After treatment, surviving clones can repopulate the tumor. And finally, metastatic lesions may differ from primary tumor genetically. That means treatment chosen from the original biopsy may no longer be optimal. Therefore, heterogeneity is not just a biological curiosity. It directly affects patients' outcome. So after heterogeneity, let's look into drug resistance. Drug resistance is perhaps the clearest example of tumor evolution. Imagine a tumor with 99% sensitive cells and 1% resistant cells. So when the therapy begins, sensitive cells die, resistant cells survive, competition is removed, and resistant cells expand. Eventually, the tumor returns, now dominated by resistant clones. This resembles antibiotic resistance in bacteria. The treatment did not create resistance from nothing. Often, resistant cells already existed in small numbers and were selective. Thus, therapy itself can unintentionally shape tumor evolution. So now let's look into the types of drug resistance. There are three main forms of resistance: intrinsic, acquired, and adaptive. In intrinsic resistance, the tumor is resistant from the start due to the pre-existing mechanisms. While in acquired resistance, initially sensitive tumors later evolve resistance after treatment exposure. And in adaptive resistance, cells temporarily reprogram themselves under stress without permanent mutations. Examples include activation of alternative pathways, dormancy states, or epigenetic changes. This shows resistance can arise through both genetic and non-genetic evolution. Now let's go through one example, EGFR mutant lung cancer. A classic example comes from non-small cell lung cancer. Some patients carry activating mutations in EGFR. They often respond dramatically to EGFR inhibitors. Tumors shrink, symptoms improve, and progression slows. However, many patients later relapse. A common cause is the emergence of T790M mutation, which alters the drug binding site. This resistant clone expands under treatment pressure. Newer drugs were then developed specifically to target T790M. This cycle beautifully illustrates co-evolution between medicine and cancer. Now let's go through tumor microenvironment as selective pressure. Cancer cells do not evolve alone. They interact with the tumor microenvironment, including immune cells, fibroblasts, blood vessels, extracellular matrix, nutrient gradients, and low oxygen regions. These factors create selective pressures. For example, low oxygen may favor cells that switch metabolism. Immune attack favors cells that hide antigens. Dense tissue favors invasive clones. Thus, evolution is shaped not only by mutations, but also by ecological context. Now let's look into the cancer development process. Metastasis is the spread of cancer to distant organs and is responsible for most cancer deaths. But metastasis is inefficient. A cancer cell must detach from the primary tumor, invade surrounding tissue, enter circulation, survive immune attack, exit blood vessels, and colonize a new organ. Most cells fail. Only rare, highly fit clones succeed. This makes metastasis an evolutionary bottleneck, selecting especially aggressive cells. So, how do we track tumor evolution today? Modern technologies now allow us to observe evolution in real time. Single cell sequencing, liquid biopsy, ctDNA monitoring, and artificial intelligence are helping us to track evolution. Let's start with single cell sequencing. It measures mutations and gene expression cell by cell. In liquid biopsy, it detects tumor DNA fragments in blood, while in ctDNA monitoring, it tracks emerging resistance mutation before scans show relapse. And AI, the artificial intelligence, analyzes the large data sets to predict progression and treatment response. These tools are shifting oncology from reactive care to proactive evolutionary management. From the beginning of this lecture, we are seeing that cancer is continuously evolving. So, if the cancer evolves, treatment must also evolve, right? So, now let's look into new therapeutic strategies. Let's start with combination therapy. In combination therapy, it attacks multiple pathways simultaneously to reduce escape routes. Then comes the sequential therapy. It uses changing strategies over time based on tumor adaptation. Then there comes the adaptive therapy. Instead of maximum killing, maintain a stable sensitive population that suppresses resistant clones. And finally, precision oncology. Tailored treatment based on each tumor molecular profile. These approaches aim not just to destroy tumors, but to steer their evolution. Now, let's look into the future perspective. Cancer as a chronic ecosystem. The future may treat cancer more like chronic ecological management than total warfare. Rather than trying to eliminate every cell immediately, which often selects resistance, we may control populations strategically over time. This could convert some cancers into manageable long term diseases. The key message is simple: to defeat cancer, we must predict how it adapts. So, now let's summarize the whole long lecture. There are some key takeaways, starting with cancer evolves through mutation and natural selection. Tumor contains diverse competing clones. Heterogeneity drives relapse and metastasis. Drug resistance is evolution under therapy pressure. And finally, future oncology must become adaptive, personalized, and evolutionary. So, as I conclude, I would just say cancer is not just a genetic disease, it is an evolving biological system. The more deeply we understand its rules of adaptation, the better we can design therapies that stay one step ahead. Thank you everyone for listening to this whole lecture patiently. I hope you all liked it. Thank you for your attention.
Thank you, Aditi, for that insightful lecture. Our next lecture will be delivered by Shreya Das, who comes from a background in biological sciences and tissue engineering for regenerative medicine. Her lecture is titled The Hidden Driver: Tumor Microenvironment Crosstalk, Angiogenesis, and 3D Spatial Mapping. Over to you, Shreya.
Good morning, afternoon, or evening, everyone. Thank you for being here today. My name is Shreya Das, and today I'm going to present my lecture on the hidden driver, tumor microenvironment crosstalk, angiogenesis, and 3D spatial mapping. I want to make a direct argument from the very first slide. Cancer progression is not dictated by malignant cells alone. It is sustained by the ecosystem those cells construct around themselves and by the spatial logic of that ecosystem. If we keep thinking about cancer as a bag of mutated cells, we will keep missing why therapies fail, why immune responses stall, and where resistance actually lives. That is the reframe we are building today. The tumor microenvironment is not the context of cancer, it is basically the driver. So let us begin. Tumors engineer survival niches. So let me start with a deliberate provocation. Consider this. We can sequence a tumor's genome, we can identify the driver mutations, find actionable targets, and deploy precision therapies. And yet, in cancer after cancer, we still watch patients develop resistance. We still watch immune cells fail to penetrate the tumor. We still struggle to explain why the same mutation leads to different outcomes in different patients. The reason in many cases is sitting on this slide. Tumors do not simply grow. They actively engineer survival niches. They recruit and reprogram the cells around them, fibroblasts, immune cells, endothelial cells, and build a microenvironment that serves malignant persistence. And critically, therapy failure is often a microenvironment problem before it even becomes a genomic problem. Resistance frequently does not require a new mutation. It can emerge from stromal signaling, immune exclusion, or the physical architecture of the tumor boundary. The decisive biology here is not just which mutations are present, it is rather like what those mutated cells signal to their neighbors, which neighborhood they create, and how that local organization shapes the clinical behavior. This is the conceptual shift this lecture asks you to make from tumor cell biology to tumor ecosystem biology. Let me quickly orient you to how this lecture is going to be structured because the three modules are deliberately sequenced to build towards a single integrative conclusion. Module 1 establishes the tumor microenvironment as the hidden driver of immune evasion. Module 2 moves into hypoxia and angiogenesis because tumor communication is not sufficient on its own to explain progression. The microenvironment is also physically and metabolically restructured. Oxygen sensing, vascular remodeling, and metabolic reprogramming are tightly integrated through H1F1 alpha. And module 3 asks what changes when we can see all of this in 3D space. Spatial transcriptomics is not just a better sequencing technology, it is a different way of asking the question. Rather, the one that preserves the geography of biology and reveals that resistance is not uniformly distributed but even spatially organized. However, the core theme threading all the three modules is that the local biology determines the clinical outcome. Now, let us come to module 1. The tumor microenvironment is introduced in many textbooks as a context, that is the cellular neighborhood in which the malignant cells reside. That framing is way too weak. It misses the biology. The TME is better understood as a distributed regulatory system. It is composed of malignant cells, cancer-associated fibroblasts, often short-termed as CAFs, immune cell populations including T cells and macrophages, endothelial cells forming the vasculature, and the extracellular matrix, that is the ECM, scaffolding all of them together. And critically, each of these components is not just present, it is actively signaling to each other component. The CAF deserves special emphasis here because they are not just passive bystanders. In fact, the more we study them, the clearer it becomes that they are among the most active organizers in the entire tumor microenvironment. They secrete cytokines that rewire immune behavior, lay down extracellular matrix that physically excludes the T cells, and cross signal with tumor cells to reinforce the stromal architecture and inflammatory programs. What this means clinically is that the TMA is not just a physical barrier between a therapy and its target. It is rather a functional regulatory layer that can override therapeutic pressure, redirect immune responses, and sustain malignant behavior even when the tumor cells themselves are under attack. Let me now take you through the three mechanistic pillars of how immune evasion is built inside the tumor microenvironment. The first is the TGF beta signaling. CAF-derived TGF beta is one of the most powerful immune exclusion signals in solid tumors. It acts on multiple levels simultaneously. It rather suppresses the T cell activation and proliferation. It promotes the differentiation of regulatory T cells that further dampen the immune responses and drives the deposition of dense ECM that creates a physical barrier, preventing the cytotoxic T cells from penetrating into the tumor core. The result is what the pathologists call an immune excluded phenotype, that is, the tumor inflating lymphocytes that are abundant at the perimeter but absent at the tumor stroma boundary. This boundary, as we'll see in module 3 as well, is one of the most clinically important zones in the tumor microenvironment. The second comes as the interleukin 6 and the STAT3 signaling. Interleukin 6 is secreted by both the tumor cells as well as the CAFs and acts on the myeloid cells, that is the macrophages and the myeloid derived suppressor cells, driving them towards a pro-tumor anti-inflammatory phenotype. STAT3 activation inside these myeloid cells sustains a feed-forward inflammatory program that does not kill the tumor but instead reinforces the suppressive stroma. The tumor benefits from inflammation while being protected from anti-tumor immunity. The third comes the INF gamma and the PD-L1 axis. This is the most clinically familiar. When cytotoxic T cells do penetrate the tumor and release interferon gamma, the tumor cells respond by upregulating PD-L1 on their surface. PD-L1 then binds PD-1 on the T cell, inducing dysfunction and progressive exhaustion. What is conceptually important here is that this adaptive resistance, the tumor is using the immune response itself as a trigger to switch on its own immune checkpoint. The stronger the immune attack, in many contexts, the stronger the adaptive counter signal. So now, what does this all add up to? Immune evasion is not a diffuse passive phenomenon. It is rather locally constructed, actively maintained and rebuilt whenever it is disrupted. That is why targeting one signal is often insufficient. The loop must be interrupted at multiple loads. I want to slow down at this slide and teach directly from the figure because this diagram contains the logic of the entire first module. We have got three subcellular actors in a triangle: the tumor cell at the bottom, the cancer associated fibroblast on one arm, and the immune cell compartment on the other. Every connection between these actors is bidirectional. There is no one-way command. The tumor cell sends TGF beta and CXCL12 to the calf. The calf responds by reinforcing the ECM and feeding back the stromal remodeling cues. The calf sends IL-6 and CCL2 to the immune compartment. The immune cells send INF gamma signals back. The tumor cell responds to the INF gamma by upregulating PDL1, which again then disables the T cell. The T cell disables itself by engaging a checkpoint. The tumor is created. Now, notice what is at the center of this loop. Not a cell, rather a feedback system. The tumor survives not because it is individually stronger than any therapeutic intervention, but because it has outsourced its control to the ecosystem around it. Any one node that is targeted is stabilized by the others. That is why network-aware intervention matters. Single agent checkpoint blockade, for example, can release the PDL1 brake. But if the TGF beta mediated exclusion barrier from the CAF compartment remains intact, T cells cannot reach the tumor core even then. You have addressed one arm of the triangle and left the others functionally dominant. Hence, the translational insight says that combination strategies must be designed within this network logic in mind. Now we are moving into module 2. If considering the module 1 was about communication, that is how the cells signal to one another to sustain a suppressive microenvironment, module 2 is about the physical and metabolic conditions that amplify this communication. Specifically, how hypoxia rewires the entire tumor biology through the HIF1 alpha signaling and what that means for vasculature, metabolism, immunity, and evasion. As a tumor grows beyond the reach of its existing blood supply. Typically, when the tumor diameter exceeds roughly 1 to 2 millimeters, it outgrows a passive diffusion of oxygen from the surrounding vessels. Regions of the tumor begin to experience oxygen concentrations below 1%. This is called hypoxia. The classical view of hypoxia in tumor biology was that it was a condition rather than an environmental stress that the cancer cells had to tolerate. However, that view is incomplete. Hypoxia is better understood as a transcriptional state. When the oxygen falls below a threshold, HIF1 alpha, that is the hypoxia inducible factor, gets stabilized and it enters the nucleus, where it drives the expression of hundreds of downstream target genes. That transcriptional program touches virtually every major adaptive process in the tumor. VEGF is induced, driving angiogenesis. GLUT1 and glycolytic enzymes get upregulated, shifting metabolism away from oxidative phosphorylation. Immune modulatory genes are activated that promote the T regulatory cells differentiation and the M2 macrophage polarization. Genes supporting cell motility and matrix invasion get turned on. Hypoxia, in other words, is not one event in tumor progression. It is rather the master switch that synchronizes angiogenesis, metabolism, immune suppression, and invasion into one integrated adaptive program. And the critical clinical point is that because hypoxia selects for cells that can tolerate and exploit it, it also becomes an evolutionary filter. The cells that survive and proliferate in hypoxic zones are disproportionately the cells with aggressive, stress tolerant, and therapy resistant phenotypes. Now, let us make the mechanism precise because it is one of the most important regulatory accesses in all of cancer biology and it is worth understanding at the molecular level. Under normal oxygen conditions, that is called the normoxia, the HIF1 alpha is continuously synthesized and continuously degraded. This degradation is driven by a set of enzymes called prolyl hydroxylase dominant proteins or the PHDs, which hydroxylate HIF1 alpha at two specific proline residues. That hydroxylation marks it for capture by the VHL ubiquitin ligase complex, which attaches a ubiquitin chain that targets the HIF1 alpha for proteasomal destruction. The protein has a half-life of under 5 minutes under normoxia. It essentially never gets accumulated. However, Under hypoxia, the PhD enzymes require oxygen as a co-substrate, and when the oxygen level drops, the PhD activity falls. HIF1 alpha is no longer hydroxylated, it rather accumulates rapidly in the cytoplasm, dimerases with its partner HIF1 beta, and the dimer translocates to the nucleus where it binds to the hypoxia response elements in the genome. The resulting transcriptional output includes VEGF, which now recruits new blood vessels, GLUT1, and other glycolytic enzymes CA1X, which regulates pH in the acidic hypoxic niche, angiopoietins, and even erythropoietin. Now, here is the paradox at the heart of tumor angiogenesis. Hypoxia induces VEGF, VEGF induces blood vessel growth, but the vessels that are formed in VEGF-rich tumor microenvironment are structurally abnormal. They are leaky, irregularly branched, poorly perfused, and chaotic. They do not restore normal oxygenation. So the tumor continues to be hypoxic despite having induced vascular growth. The angiogenic switch is on, but the oxygen problem that triggered it is never resolved. That is why chronic hypoxia persists in advanced solid tumors despite extensive neovascularization. I want to now show you why hypoxia is not simply a vascular problem, rather it's a full systems problem that simultaneously rewires three major downstream programs. The first is the metabolic reprogramming. Under HIF1 alpha, tumor cells massively upregulate glycolytic activity, shifting energy production towards the Warburg pathway. This generates ATP under oxygen poor conditions, but it even generates large quantities of lactate. Lactate acidifies the extracellular environment and critically is itself a signaling molecule that suppresses the T cell activity and supports the immune evasion. The acidic lactate rich hypoxic niche is therefore metabolically self-reinforcing. The second comes the immune suppression. Hypoxia directly promotes the expansion of regulatory T cells and drives macrophages towards the M2 anti-inflammatory phenotype. Lactate accumulation further impairs the cytotoxic T lymphocyte function. So the same metabolic reprogramming that sustains tumor cell viability under oxygen stress also actively dismantles the local immune surveillance. The third program is invasion and metastasis. HIF1 alpha drives the epithelial to mesenchymal transition, the process by which the tumor cells downregulate the adhesion molecules, upregulate the mesenchymal markers, and acquire a migratory phenotype. Matrix metalloproteinases are induced, which degrade the basement membrane. The CXCR4-SDF1 chemokine axis gets activated, guiding the cells towards the blood and lymphatic vessels. All of these are HIF-driven, and all of them converge into to promote dissemination. What is now most important to understand is that these three programs reinforce each other. The metabolic niche created by glycolytic reprogramming suppresses immunity. The immune suppression allows invasion to proceed unchecked. The invasion creates new metastatic niches that are themselves rapidly hypoxic. HIF is a master regulator that holds all of this together. We are now entering module 3, and this in many ways the module that changes what we can do with everything we have just learned. Module 1 and 2 gave us the molecular biology, whereas module 3 now gives us the spatial resolution to actually see where that biology is organized and where it should be targeted. For most of the molecular biology's history, the methods we've used to measure gene expression destroyed the very spatial relationships that generated those expression states in the very first place. With bulk RNA sequencing, we homogenize a tissue sample. We get an average transcriptional profile across millions of cells, many of them different cell types, many of them in different zones of the tumor, all of them collapsed into one data point. The geography is gone. With single-cell RNA sequencing, we made enormous progress on cellular heterogeneity. We can now distinguish from a single tumor dozens of distinct cell states like the exhausted T cells, CAF subtypes, hypoxic tumor clusters, myeloid polarization states. But to generalize and to generate single-cell data, we need to first dissociate the tissue. We break apart the physical architecture. We lose the spatial relationships between those cell states. Here is why that matters. A tumor cell in direct contact with a TGF-beta secreting CAF behaves very different from the same tumor cell that is 10 micrometers away from the nearest calf. Single cell data can tell us that both the cell states exist. However, it cannot tell which cells are actually adjacent, and adjacency is biology. Spatial transcriptomics preserves the tissue architecture while measuring gene expression in situ. We can now ask not just what is expressed, but where and critically adjacent to whom. And that is a completely different question, and it is this question that the module 3 is built around. What does spatial transcriptomics actually reveal when we apply it to solid tumors? The first finding is that it is consistently emerging, like the ligand receptor interactions, that is the molecular conversations between the cells are not uniformly distributed across the tumor. They are only spatially clustered. When you map the CXCL12 and the CXCR4 signaling between CAFs and the T cells, you do not find it everywhere. Rather, you find it concentrated at the tumor stroma boundary. That boundary is not just a histological feature, it is rather a signaling hotspot where some of the most important biology governing immune exclusion and therapy resistance is organized. The second major finding is that the exhausted T cells are spatially restricted. They are not simply present throughout the tumor in a dysfunctional state. They are concentrated as specific marginal zones surrounded by suppressive stroma, physically barred from advancing into the tumor core by CAF-laid matrix. Understanding that geography tells us not just that the T cells exhausted, but where they are stalled and what is stalling them. Third, therapy resistance is frequently regional and not uniform. A tumor does not develop uniform resistance to checkpoint blockade. It develops resistant niches, specific microregions with distinct stromal compositions, distinct immune cell positioning, and distinct ligand receptor environments that shield subpopulations of tumor cells from the therapeutic pressure. In colorectal cancer, spatial organization of the tumor stroma boundary has already been linked to differential response to immune checkpoint blockade, and that is clinically actionable spatial biology. That is the promise this technology delivers on. Let me close with the synthesis that all the three modules have been building toward. The tumor microenvironment is not the background. It is a communication network actively constructed, continuously maintained, and populated by cellular actors that have been co-opted, reprogrammed, or recruited to sustain malignant behavior. Treating the TME as a bystander is a clinical and even a conceptual mistake. Hypoxia is not a passive condition of undervascularization. It is a progression engine, a transcriptional master switch that simultaneously drives angiogenesis, metabolic adaptation, immune suppression, and invasive behavior through the HIF1 alpha axis. It synchronizes and amplifies all of the stromal interactions as we described in Module 1. Spatial transcriptomics is not just a better map of known biology, it is a next generation lens that reveals where the biology is organized, where suppressive niches are built, where exhausted T cells are stranded, where the resistance geography of the tumor actually lives. If genomics told us what the tumor is, what it has mutated, what drivers it carries, what vulnerabilities it encodes, then spatial oncology is the beginning to tell us something different, where the tumor survives, where it resists, and where we should strike next. The future direction of this field is rational combination therapy, designed around spatial biomarkers of resistance therapy that is not just molecularly targeted but ecosystem aware. That is the translational frontier that this lecture has been building toward. Thank you. I'll be happy to take any questions.
Thank you, Shreya, for that excellent lecture. Our next lecture will be delivered by Shri Deepti B, who holds a master's degree in biotechnology. Her lecture is titled Cancer Progression. Welcome, Deepti.
Hello everyone. So today I'll be talking about cancer progression. And to understand progression properly, we first need to understand the hallmarks of cancer, which is conceptual framework introduced to explain how normal cells transform into malignant ones. Now, cancer is not just one disease, right? And there are over 100 types. But despite this diversity, cancer cells share certain core biological capabilities. Initially in 2000, six hallmarks were proposed: sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastasis. Later, this model evolved. By 2011, additional hallmarks like immune evasion, tumor promoting inflammation, genome instability, and metabolic reprogramming, which I will discuss in the further slides, they were added. Now, in coming to the most recent one, which is 2022, newer dimensions such as epigenetic reprogramming, microbiome influence, senescence, and phenotypic plasticity were included. So overall, what this tells us is cancer is not caused by a single change, but by multiple coordinated alterations in cell physiology. Now, moving on to how cancer actually begins. One key concept here is tumor clonality, which means most tumors arise from a single abnormal cell. But that doesn't mean that one mutation is enough. Cancer development is actually a multi-step process. So now, as you can see in the image as well, the first thing is tumor initiation, where a genetic mutation occurs. Then cell proliferation, where that mutated cell starts dividing, followed by tumor progression, where additional mutations accumulate. As you can see here, the orange and the white, these are additional mutations. And finally, clonal selection occurs, where the most aggressive cells dominate. So, cancer is essentially an evolutionary process happening inside the body. To understand this better, let's take colon cancer as an example. It starts with a single epithelial cell undergoing abnormal proliferation. This progress is like step by step. So, first a small cluster forms, then a benign adenoma, which is non-cancerous. Then this grows into intermediate and then large adenomas. Eventually, cells invade through the basal lamina, becoming malignant. At this point, the cancer gains the ability to invade surrounding tissues, enter blood and lymphatic vessels, and metastasize to distant organs. So, what's important here is cancer doesn't suddenly appear as malignant. It gradually evolves from benign to invasive stages. Now, the question is what drives this progression at the genetic level? Cancer cells show multiple chromosomal abnormalities. First is aneuploidy, where there is an abnormal number of chromosomes, which can lead to either gene amplification or gene loss. Second is gene amplification, where certain oncogenes are overexpressed due to multiple copies. Third are deletions, where tumor suppressor genes may be deleted or lost. And finally, loss of heterozygosity, which disrupts normal gene regulation. So, overall, these genetic alterations disturb the balance between cell growth, cell death, and DNA repair. And that very imbalance is what drives malignancy. Now, this is a very important concept, which is the Warburg effect or metabolic reprogramming in cancer. So normally cells metabolize glucose like this. So in the presence of oxygen, oxidative phosphorylation occurs, uh, where 36 molecules ATP is produced. And in the absence of oxygen, glycolysis occurs where only two ATP molecules are produced. But cancer cells behave differently. Even in the presence of oxygen, they prefer aerobic glycolysis, converting glucose into lactate. This seems inefficient, but it actually benefits cancer cells because uh, it provides a rapid ATP and, more importantly, it supplies intermediates for biosynthesis. We also see increased glucose uptake via GLUT1 and GLUT3, increased glycolytic enzymes like hexokinase and PKM2. In, we see increased lactate production, which acidifies the tumor environment, which is basically beneficial for the tumor. So the key idea over here is cancer cells prioritize growth and biomass production over energy efficiency. Now let's connect this to hypoxia, which is very common in tumors. As tumors grow rapidly, oxygen supply cannot keep up with demand. Also, tumor blood vessels are abnormal and inefficient. This leads to low oxygen conditions or hypoxia. Hypoxia has several important effects. It activates HIF1, which is a master regulator. It induces epithelial to mesenchymal transition, which is EMT, increasing metastasis. It promotes therapy resistance and it alters multiple signaling pathways like PI3K, MAPK, and NF-kappa beta. HIF1 in particular drives metabolic reprogramming, survival pathways, and even drug resistance. So, hypoxia doesn't just result from cancer, it actively drives cancer progression. So, cancer is not just about tumor cells, right? It's about the tumor microenvironment, which includes immune cells, stromal cells like fibroblasts, blood vessels, ECM, and secreted factors. Together, these components create a supportive niche for tumor growth. One important feature is gradient. Um, so now, uh, as you can see in over here, near the region near blood vessels, there's normal oxygen, and in the deeper regions, we can see hypoxia and acidity. And this was also depicted in the previous slide. So due to the Warburg effect, lactate accumulates, making the environment acidic. Also, the tumor microenvironment becomes immunosuppressive, where there is increased IL-10, TGF-beta, and decreased pro-inflammatory cytokines. There are also physical barriers like dense extracellular matrix, which prevent drug penetration. So, essentially, the tumor microenvironment protects the cancer and helps it survive and spread. Now, understanding all of this has major implications for treatment. Targeting cancer cells alone is not enough. Effective therapy needs to target the tumor vasculature using anti-angiogenic drugs, the immune system, which uses immunotherapy, and the cancer cells themselves using cytotoxic or targeted therapies. This is why modern cancer treatment focuses on combination therapies because cancer progression is complex, which involves genetics, metabolism, hypoxia, and microenvironment. So the treatment must also be multi-targeted. So, the future of cancer therapy lies in integrating all these approaches together. Thank you.
Thank you, Deepti, for that informative lecture. With that, we conclude theme 1 of today's lecture series. We will now take a 10-minute break and resume shortly with theme 2. Please rejoin us in 10 minutes. Thank you, everyone. Welcome back, everyone. We now begin Theme 2, where today's lectures will explore the latest advances in cancer therapeutics, from molecular targeting strategies to precision medicine approaches. I welcome my co host, Mr. Aditya Pawar. Aditya, over to you.
Our first lecture in this session will be delivered by Ashley Varghese. Her lecture is on advances in cancer therapeutics from molecular targets to precision medicine. Ashley, over to you.
Good afternoon. I am Ashley Vargas. Today I will be discussing advances in cancer therapeutics from molecular targets to precision medicine. Cancer is not a single disease, but a collection of disorder given by genetic and molecular abnormalities. Because of this complexity, treatment strategies have evolved significantly, and that's what we will be discussing today. The evolution of cancer treatment. Let's start with how cancer treatment has evolved. Initially, treatment relied on three major approaches: surgery, that is physically removing the tumors, radiotherapy, using ionization radiations to damage the DNA, chemotherapy, using cytotoxic drugs. These methods were effective but non specific. Chemotherapy, for example, targets all rapidly dividing cells, not just cancer. Over the last 25 years, we have moved into a molecular era where treatment is based on tumor biology. This evolution includes targeted therapy, blocking specific molecules, immunotherapy, activating the immune system, and precision medicine, customizing treatment based on the genetic profile. So the shift is from one size fits all to individualized treatment. Despite their importance, traditional therapies have major drawbacks. That is non specific toxicity. Chemotherapy affects bone marrow, causing anemia and infection. It affects the GI tract, causing nausea and diarrhea. It also affects the hair follicles, resulting in hair loss. This significantly impacts the patient's quality of life. The second thing is drug resistance. Tumors are genetically unstable and can evolve. Mechanism includes mutation in drug targets, increased drug efflux pumps, and activation of alternative pathway. This leads to treatment failures over time. Reoccurrence. Even after treatment, some cancer cells may survive. These are often called cancer stem cells. They resist therapy, regenerate tumors, lead to aggressive relapses. So, clearly, more precise and durable therapies were needed. So, why there is a need for targeted therapy? Because it focuses on specific molecular abnormalities in cancer. Unlike chemotherapy, it interferes with specific proteins or pathways essential for tumor survival. For example, it may affect the receptors, that is EGFR or HER2, which is overly expressed in breast cancer. These receptors trigger uncontrolled growth when activated, or it may affect signaling pathways like uh, which affects the cell proliferation and survival and metabolism. In cancer, these pathways are overreactive, or it may affect the angiogenesis. Tumors need blood supplies to grow. By blocking certain pathways, may starve the tumor. So, targeted therapy is like hitting the bull eye instead of shooting blindly. Let's look at some of the breakthrough drugs that changed oncology. This one is Gleevec. It targets the BCLR ABL fusion protein in CML. This protein is a consequently active tyrosine kinase. This drug inhibits this enzyme, stops the cancer proliferation, and it in turn increases the survival rate of the patient from 20% to nearly 90%. Next drug is Herceptin, which targets HER2 receptor, which is very aggressively expressed in cancer, especially in breast. This drug blocks the signaling and also triggers immune destruction, and which in turn results in 50% reduction in recurrence. Last drug targets the EGFR mutation in lung cancer, works only in patients with specific mutations, early example of precision medicine. These drugs prove that if you're understanding molecular biology well, it will give a better therapy outcomes. Immunotherapy. Now moving to one of the most exciting areas, immunotherapy. Instead of targeting cancers directly, this approach empowers the immune system. Normally, our immune system can detect abnormal cells, but cancer cells can hide from immune system and suppresses immune responses. Immune therapy removes these barriers. These types include the checkpoint inhibitors, the CAR T therapy, and the monoclonal antibody. The goal is reactivate immune surveillance. Now we will discuss two of the most important revolutionary breakthrough in cancer immunotherapy. Immune checkpoint inhibitors and CAR T cell therapy. Both approaches aim to enhance the immune system, but they do so in fundamentally different way. First one is the checkpoint inhibitors. Our immune system, especially T cells, is capable of recognizing and destroying cancer cells. However, to prevent excessive immune reaction, T cells have regulatory mechanism called immune checkpoints. Two important checkpoints are PD-1, that is programmed cell death protein 1, present on T cells, as well as cytotoxic T lymphocyte associated protein 4, also on T cell. Their role is to prevent overactivation of the immune system and avoid autoimmunity. And how does cancer escape the system? Actually, cancer cells exploit the system. They express a ligand called PDL1, which binds to PD1 on T cells. This sends an inhibitory signal to the T cell. The T cell becomes inactive or exhausted. Cancer cells escape immune destruction. So, mechanism of the drugs. Checkpoint inhibitors block this interaction. Example include two of the drugs which block PD1 and also another drug which is called Impilum, which blocks CTLA4. When these drugs block the checkpoints, T cells are reactivated, cytotoxic activity increases, and tumor cells are attacked and destroyed. Clinical impact of these therapies has is it have shown incredible outcomes, that is durable response in melanoma, improved survival in lung cancer, long-term remission in some patients. Unlike chemotherapy, responses can last for years. But there are some side effects also. Since the immune system is activated, it can attack normal tissue also. And the next important breakthrough is CAR T cell therapy, which is a form of adaptive cell transfer therapy and is highly personalized. It involves genetically modifying a patient's own T cells to better recognize the cancer cell. CAR T therapy involves several steps, that is T cell collection. We collect the T cells from the patient and a gene encoding a chimeric antigen receptor CAR is introduced into T cells. This receptor combines the antigen recognition domain like antibody and T cell activation domain. The modified T cells are grown in large numbers in the lab, and these engineered T cells are infused back into the patients. Once inside, they directly recognize the tumor antigen and kill the cancer cells. Unlike normal T cells, CAR T cells do not require antigen presentation to MHC. This allows faster recognition, stronger cytotoxic responses. They release perforin and granzymes, leading to apoptosis of cancer cells. So, the clinical success of CAR T includes the 80% remission rates in pediatric acute lymphoblastic leukemia, success in B cells lymphoma, and multiple myeloma. It is especially effective in hematological cancers. There is some limitations also through CAR T. However, like CRS, that's a cytokine release syndrome, that is massive cytokine is released, which will lead to fever, hypertension, and organ dysfunction. And also cases of neurotoxicity has also been responded. Moving forward, One of the most important advances in cancer therapy is precision medicine. It focuses on tailoring treatments based on genetic and molecular profile of an individual patient's tumor. Traditionally, patients with same cancer receive the same treatment. However, tumors are genetically heterogeneous, meaning patients respond differently to the same drug. Precision medicine addresses this by selecting therapies based on specific mutations. The first component of the precision medicine is tumor genomic sequencing. Techniques like NGS identify driver mutations, and these mutations act as actionable targets, allowing user specific inhibitors. Second one is liquid biopsy. This involves detecting circulating tumor DNA in blood. It's non invasive and helps monitor treatment responses and detects early relapse. And also, it helps in identifying resistant mutations. The third important component is companion diagnostics. These are tests used before the treatment to ensure the drug's potential. Example HER2 testing before the drug as well as EGFR. This avoids unnecessary treatment and improves outcome. So why does such testing matters or why does precision medicine matters? Because it leads to better efficiency, reduced toxicity and more personalized care. It shifts cancer treatment from one size fits all approach to individualized therapy. In summary, precision medicine treats cancer based on its molecular characteristics rather than its location, making therapy. Another major advancement in cancer therapy is the use of nanotechnology for drug delivery. It focuses on improving how drugs reach the tumor, making treatments more efficient and less toxic. So, we have seen problems with conventional therapy. To summarize, the traditional chemotherapy has several limitations like drug solubility is very poor, non specific distribution throughout the body, and high systemic toxicity. As a result, only a small fraction of drug reaches the actual tumor. So, nanotechnology involves using nanoparticles, typically 1 to 100 nanometers in size, to deliver drugs. These particles can be engineered to carry therapeutic agents, protect the drug from degradation, and release drugs in a controlled manner. So its mechanism include enhanced permeability and retention effects, also known as EPR effect. So tumors have a leaky blood vessel and poor lymphatic drainage. This allows nanoparticles to enter tumor tissues easily, accumulate more than a normal tissue. This is called EPR effect and it improves the drug targeting. Lipid nanoparticles, these are widely used nanocarriers. These are important for delivering mRNAs, siRNAs, gene editing tools. A well-known example in the is the use in mRNA vaccines. Now similar therapies are used in cancer therapy. So advantages of nanotechnology involves improved drug delivery to tumor, reduced size effects on normal tissue, better drug stability, possibility of targeted and controlled disease. Overall, it enhances both efficiency and safety of cancer treatment. To summarize, nanotechnology uh, transforms conventional chemotherapy into more targeted and efficient system, making it a key component of future therapeutics, especially for cancer. From translational perspective, nanotechnology bridges the gap between molecular discoveries and clinical application by improving drug delivery system. While recent advances in cancer therapy are highly promising, they also come with several important challenges that need to be addressed. First one is drug resistance. One of the biggest challenges is drug resistance. Cancer cells are genetically unstable and can evolve under treatment pressure. They develop resistance through new mutation in a drug target, activation of alternative signaling pathways, increased drug efflux. As a result, therapies that initially work may become ineffective over time. This is why combination therapies are often needed and often used. Cost and accessibility. Another major issue is the high cost of advanced therapies. For example, CAR-T alone can cost extremely high. Targeted therapies and biologics are also very expensive. This creates inequality in access, especially in developing countries. The affordability is a major concern in translating these advances globally. The third one is toxicity and side effects. Although newer therapies are more targeted, they are still very, very back in providing the maximum optimization uh, while significantly reducing side effects. Examples include immunotherapy related toxicity. That is, it can trigger an autoimmune reaction, inflammation of organs, especially liver or colon. And it also induces toxicity. That is, it can lead to CRS, as discussed earlier, that is cytokine release syndrome, which can lead to fever or severe allergic reaction. And also, it is reported in some cases to create neurotoxicity. These can sometimes be very severe and require very careful monitoring. To summarize this entire slide, while modern cancer therapies have improved outcomes, significantly challenges like um, resistance, cost, and toxicity must be addressed to make these treatments more effective and accessible. Addressing these challenges is essential to ensure that scientific advancements translate into real world clinical benefits for all patients. To conclude, cancer therapeutics have undergone a remarkable transformation from non specific treatments like chemotherapy to highly advanced approaches such as targeted therapy, immunotherapy, and precision medicine. Today, we are not just treating cancer based on its location, but its on molecular as well as genetic profile, allowing for more personalized and effective care. Technologies like nanotechnology and genomic sequencing are further enhancing treatment precision and reducing side effects. However, challenges such as drug resistance, toxicity, and high cost remain, and addressing these will be critical for future progress. Overall, this field is moving forward for more precision and patient specific, targeted, and efficient approach, offering real hope for improved survival and quality of life. The future of cancer therapy lies not just in killing cancer cells, but in understanding them at their deepest molecular level. Thank you.
We now move to the next lecture, again by Aditi Dubey. Her lecture focuses on from one size fits all to precision oncology, modern advances in cancer therapy. Aditi, please continue.
Once again, a very good afternoon to everyone. I am Aditi Dubey, and today it's a privilege to present a topic that reflects one of the most remarkable transformations in modern medicine, that is, from one size fits to all to precision oncology, modern advances in cancer therapeutics. Cancer treatment has changed dramatically over the past few decades. Historically, many patients with the same type of cancer were treated with the same therapies. However, we now understand that two tumors arising in the same organ may behave very differently because their molecular biology is different. This realization has led to a major shift from generalized treatment strategies toward therapies designed around the unique characteristics of each patient's tumor. Today, I will discuss how cancer therapies evolved from surgery, radiation, and chemotherapy to targeted therapies, immunotherapy, genomics driven medicine, and artificial intelligence supported care. So, now let's start with the historical evolution of cancer treatment. The history of cancer treatment can be divided into major eras. The earliest pillar was surgery, aimed at physically removing tumors. Later came radiation therapy, which uses ionizing radiations to destroy malignant cells. Then, in the 20th century, chemotherapy became a major breakthrough. Cytotoxic drugs improved survival in many cancers and remain essential today. However, chemotherapy was often broad rather than selective. In the modern era, new pillars emerged, like target therapy. immunotherapy, precision medicine, and data guided oncology. These newer approaches focus not just on eliminating dividing cells but on understanding the specific vulnerabilities of cancer. Why did the older model become insufficient? Because the same diagnosis does not mean the same disease biology. For example, two patients with breast cancer may have completely different molecular subtypes. One may respond well to hormone therapy, another may need HER2 targeted therapy, and another may require chemotherapy. Similarly, two lung cancers may look identical under the microscope but carry different mutations requiring different treatments. The one size fits all model created several problems: inconsistent responses, unnecessary toxicity, overtreatment in some patients, undertreatment in others, and development of resistance. Thus, clinicians and researchers needed more personalized approaches. So, what is precision oncology? Precision oncology means selecting treatment based on the unique biological features of a tumor and the characteristics of the patient. Rather than relying only on the organ of origin, treatment decisions may consider genomic mutations, biomarker status, protein expression, immune profile, prior treatment history, and real time disease monitoring. The core principle is simple: right treatment, right patient, right time. Precision oncology aims to maximize benefit while minimizing unnecessary toxicity. Now, let's look cancer as a molecular disease. Cancer is fundamentally driven by molecular alterations. These may include DNA mutations, gene amplifications, chromosomal rearrangements, epigenetic dysregulation, and abnormal signaling pathways. These changes activate oncogenes or disable tumor suppressor genes. For example, TP53, KRAS, because tumors accumulate different combinations of alterations. No two cancers are exactly alike. This is why molecular testing has become so important in treatment planning. Now, what are the landmark molecular targets? Several discoveries changed oncology forever. EGFR. EGFR mutations identify patients who may respond to EGFR inhibitors. HER2. HER2 amplifications transformed treatment in a subset of breast cancer. BCR-ABL. This led to one of the most successful targeted therapies in history. BRAF, commonly targeted in melanoma. BRCA1 and 2, important in ovarian and breast cancer, especially for PARP inhibitor therapy. These examples prove that understanding biology can directly guide treatment. So, the targeted therapies are drugs designed to inhibit specific molecules or pathways required for cancer survival. Major classes include tyrosine kinase inhibitors, monoclonal antibodies, PARP inhibitors, CDK4 or 6 inhibitors. Compared with conventional chemotherapy, targeted therapies often provide greater selectivity, better response in biomarker positive patients, improved tolerability, and oral treatment options in some cases. For many cancers, targeted therapies has dramatically prolonged survival. However, resistance remains a challenge, which I will discuss later. Now let's move to immunotherapy. Immunotherapy has changed the landscape. Instead of directly attacking the tumor, immunotherapy activates the patient's immune system. Important forms include checkpoint inhibitors, CAR T cell therapy, cancer vaccines, and combination immune strategies. Checkpoint inhibitors block molecules such as PD1 or PDL1 that tumor uses to suppress immunity. Some patients with advanced cancer have achieved durable long-term responses previously thought impossible. Immunotherapy has transformed treatment in melanoma, lung cancer, kidney cancer, and several other malignancies. So now biomarkers play a very important role. Precision medicine depends on biomarkers. Biomarkers help predict who is most likely to benefit from a treatment. Examples may include PD-L1 expression for immunotherapy decisions, MSI high status for checkpoint inhibitor sensitivity, HER2 positivity for anti-HER2 therapy, hormone receptor positivity in breast cancer, tumor mutational burden in selected settings. Without biomarkers, therapies may be used less effectively. With biomarkers, treatment becomes smarter and more efficient. Now let's dive into the role of genomic sequencing. A major engine behind precision oncology is next generation sequencing, or we call it as NGS. NGS can test many genes simultaneously from a tumor sample. It helps identify actionable mutations, rare genomic alterations, fusion genes, resistance mutations, and eligibility for clinical trials. Previously, testing genes one by one was slow and expensive. Now, one panel may evaluate dozens or hundreds of genes. This has accelerated personalized treatment decisions. After genomic sequencing, let's look into the role of AI in modern oncology. Artificial intelligence, or AI, is becoming increasingly important in oncology. AI can analyze enormous data sets beyond human capacity. Applications include reading CT and MRI scans, detecting subtle pathology patterns, predicting reoccurrence risk, suggesting treatment options, and integrating genomics with outcomes. For example, AI may help identify which patients need aggressive treatment and which patients can avoid overtreatment. Here, a point to remember and to notice: AI supports clinicians, it does not replace them. Now, in my previous lecture, I talked about liquid biopsy and real time monitoring. Let's look a little deep in this. One exciting innovation is liquid biopsy. Instead of repeatedly sampling tumor tissue, clinicians analyze blood for tumor-derived signals. This includes circulating tumor DNA, circulating tumor cells, and other tumor fragments. Benefits may include less invasive testing, detection of resistance mutation, monitoring treatment response, identifying minimal residual disease, and earlier relapse detection. This allows cancer care to become more dynamic and adaptive. So here is a clinical example. HER2 positive breast cancer. Consider breast cancer. Before targeted therapies, HER2 positive disease was associated with aggressive behavior and poorer outcomes. Then, anti-HER2 therapies such as trastuzumab changed the field. Patients experienced better response rate, long survival, and improved disease control. This is a classic example of how identifying a biomarker can completely transform prognosis. Have you ever thought, after all these advancements, why there is still resistance happening? Despite remarkable progress, resistance remains one of the greatest obstacles. Tumors may adapt through secondary mutations, activation of bypass pathways, tumor heterogeneity, drug efflux mechanisms, and immune escape. For example, a targeted therapy may initially work well, but resistant clones eventually emerge. This is why modern treatment increasingly uses combinations, sequencing strategies, and repeat molecular testing. Cancer is dynamic, so therapy must also be dynamic. Even after all the advancements, there are still some challenges in precision oncology, and these are broader ones. Let's start with cost. Sequencing, biologics, and advanced therapies may be expensive. Then comes the access. Many patients worldwide lack access to testing and specialized care. Then, interpretation. Not every mutation is clinically meaningful. Then comes the infrastructure. Precision care requires pathologists, oncologists, geneticists, bioinformaticians, and pharmacists working together. And one of the important challenges, ethics and privacy. Genomic data must be handled carefully. Scientific progress must be matched by equitable delivery. Now let's look into the future of cancer therapeutics. The future is extremely promising. Likely developments include personalized cancer vaccines, CRISPR-based therapies, AI-guided treatment planning, multi-omics integration, adaptive treatment based on serial monitoring, earlier detection using blood-based tests. We are moving from reactive medicine toward predictive medicine. In the future, treatment may be adjusted continuously as the tumor changes. So now let's summarize the whole lecture again. There are some key takeaways. First, one size fits all oncology is fading. Tumor biology now guides treatment choices. Targeted therapy and immunotherapy have transformed outcomes. Biomarkers and genomics are central to modern care. AI is accelerating diagnosis and decision making. The future of oncology is personalized, adaptive, and precise. So, now to conclude, cancer treatment has evolved from treating tumors only by location to treating tumors by molecular identity, from broad toxicity to selective intervention, from standardized care to individualized medicine. Precision oncology is not merely the future of cancer care, it is the present, and it will continue to redefine survival and quality of life for patients worldwide. Thank you very much for your attention and being patient throughout the lecture.
Thank you, Aditi. Moving on, our next lecture will be delivered by Shri Dipti B, titled Recent Advances in Cancer Therapeutics. Please go ahead.
Hello everyone. Today I'll be talking about recent advances in cancer therapeutics. Cancer treatment has undergone a massive shift over the last decade. What was once largely dependent on non-specific cytotoxic chemotherapy is now moving towards highly precise mechanism-driven approaches. Let's start with the broader picture. Traditionally, chemotherapy targeted rapidly dividing cells, which is why we saw significant toxicity. But now the focus is on precision medicine. We are targeting specific molecular abnormalities within tumor cells. Some key therapeutic classes include targeted therapies, where these inhibit oncogenic signaling pathways, immune checkpoint inhibitors, where they basically remove the brakes from the immune system, antibody drug conjugates, which is very interesting, where they deliver cytotoxic drugs directly to cancer cells, and cellular therapies like CAR T cells. So overall, The idea is more specificity and less collateral damage to our own immune cells. Now, coming to breast cancer, a major trend here is de-escalation of therapy. For example, hypofractionated radiotherapy, which is of a shorter duration, shows similar outcomes compared to longer regimens with comparable survival and toxicity profiles. Then, carboplatin omission, where we see that removing carboplatin doesn't significantly affect response rates but reduces severe adverse effects. And extended endocrine therapy, where in hormone receptor positive patients, extending therapy improves long term survival outcomes. So, here the shift is not just towards effectiveness but also towards reducing treatment burden. Now, moving to newer drugs, this is where things get more interesting. Trastuzumab deruxtecan, which is an antibody drug conjugate, shows significantly improved disease-free survival. Next, palbociclib, which is a CDK4 CDK6 inhibitor, improves progression-free survival when used as maintenance. Next, which is sacituzumab govitecan. It is particularly useful in triple-negative breast cancer. And olaparib, which is a PARP inhibitor, is especially effective in BRCA-mutated cancers. So, what we are seeing is that therapy is becoming more genotype driven rather than just phenotype driven. In gastrointestinal cancers, both pancreatic and colorectal cancers are seeing notable advances. First, coming to pancreatic cancer, we can see tumor treating fields, which is a FDA approved device based therapy using electrical fields, which improves survival when combined with chemotherapy. Preoperative combination regimens like PAXG improve event free survival. In colorectal cancer, targeted therapies like encorafenib plus cetuximab significantly improves response rates in BRAF mutant cancers. Interestingly, aggressive tumor debulking does not improve survival and may increase toxicity. So again, treatment decisions are becoming more evidence-based and selective. Looking at other gastrointestinal cancers, in hepatocellular carcinoma, combination immunotherapy improves survival significantly compared to surgery alone. In gastric and esophageal cancers, new antibody combinations are outperforming older HER2 targeted therapies. And in microsatellite stable colorectal cancer, which is usually resistant, new combinations are showing modest improvements, although toxicity is still a concern. Basically, progress is happening, but challenges remain in resistant cancers. Now, coming to kidney, bladder, and prostate cancers, in renal cell carcinoma, combining belzutifan with pembrolizumab improves disease-free survival. In bladder cancer, antibody drug conjugates combined with immunotherapy show stronger survival benefits. And in prostate cancer, enzalutamide combined with androgen deprivation therapy improves long-term survival. Also, long-term PSA screening data shows sustained reduction in mortality. Here, combination strategies are clearly dominating, is what we can see. Coming to ovarian and endometrial cancers, first, ovarian cancer, relacorilant plus nab-paclitaxel, which is a FDA-approved combination, is seen to improve both progression-free and overall survival in resistant disease. Next, pembrolizumab plus paclitaxel combinations work well in PDL1 positive cases. And ribociclib plus letrozole shows promising survival in specific subtypes. Coming to endometrial cancer, treatment is now increasingly guided by molecular profiling rather than just histology. So again, here personalization is key. In lung cancer, targeted therapies like talcetinib show very high response rates in RET mutated cancers. Other agents target HER2 and EGFR mutations effectively. In melanoma, immunotherapy continues to dominate with significant improvements in recurrence-free survival. And in rarer cancers, new targeted therapies are showing promising results. So overall, targeted and immune-based therapies are transforming outcomes even in difficult cancers. Okay, so to summarize the major trends, cancer therapeutics is increasingly moving towards personalized medicine, where molecular profiling and specific biomarkers are used to guide treatment decisions for each patient. At the same time, combination therapies are being widely adopted as they help overcome resistance mechanisms and improve overall treatment outcomes. Another important shift is the earlier use of advanced therapies, where these novel agents are now being introduced even in early stage disease to prevent recurrence and improve long-term survival. In addition, regulatory approvals are becoming more biomarker driven, which means treatments are now being approved based on specific molecular alterations rather than being limited to the organ of origin. So overall, cancer treatment is clearly evolving towards a more individualized, mechanism-based and proactive approach, and ongoing clinical trials will continue to refine and expand these strategies further. Thank you.
Thank you for that lecture. Now we invite Dr. Bindu Madhuri for the next lecture. Dr. Bindu Madhuri, the floor is yours.
Hello everyone, myself Bindu. I'm a PhD graduate in biotechnology with a specialization in drug discovery. In this session, I would like to take the opportunity to present about the advent of molecular targeted therapies against cancer towards a multi-targeting drugs. So, in this scenario, we would see some basic concepts of the cancer, how it arises, and how it evolves, and what are all the conventional chemotherapeutics that cancer is having in the control of the cancer uh, for a population betterment, and what is still lacking and how we can develop the potential drug, what are all the different ways for the development of the potential drug in control of cancer. So in the to achieve the better outcomes of the cancer control, we would like to cover some concepts of heterogeneity of the cancers and the molecular pathways that are allied in the cancer, and why we have to consider about the targeted cancer therapies. So then let us move on to the cancer basic concepts. So like here I would like to mention what at first what is cancer. Cancer is nothing but the ability of a given cell to avoid the certain hallmarks of cell proliferation and to gain the adaptability of uncontrollable cell proliferation. So in this scenario, cancer has remained as a global health burden. Why it has become a global health burden? Because according to 2022 Globocon cancer statistics, per year there are 20 million new cancer cases and 9.7 million deaths of cancer. So this itself states that cancer occurrence is high and its mortality is high. So something is influencing some disease or some abnormality is influencing the population in this level. One should focus on how it can be controlled, what all the different strategies can be developed against in the control of that abnormality, that is nothing but here it is a cancer. So cancer, as I said, its distinct feature is that the ability of the cell for its abnormal proliferation capability. So conventionally, this cancer is being treated by certain drugs such as cisplatin, doxorubicin, and paclitaxel, which their mode of action is to target the rapidly dividing cells, as it is a common feature of a cancer cell. However, it is having so many a distinct limitation is that it is not having any specificity in these drugs. These conventional chemotherapeutics are not having any specific activity, so they go and act on the normally proliferating tissues also, like bone marrow, gastrointestinal epithelium, and hair follicles, and that's how they are arising some systemic toxicity that is leading and so many adverse effects of the cancer affected individual being instead of treating the cancer. In such scenario, there are still a development is required for the better therapeutic opportunities. In such line, molecular targeted therapeutics and precision oncology comes for the patient specific interventions for the betterment of the treatment options. The advancements in multi-omics profiling and AI driven approaches supports the development of these molecularly targeted therapies and precision oncology. However, whatever the achievements are there, the tumor heterogeneity and drug resistance has remains as a major challenging for the successful development of the cancer therapeutics. So, this is the basics that we have covered in the first slide itself. How cancer comes, what are the reasons for the development of abrupted cell proliferation of the cancer. Uh, as it is very well known that cancer is a multifactorial etiology, right? So it can be influenced by the genetic mutations and the sporadic occurrence due to the mutagens in the environment or whatever the behavioral lifestyle changes. So whatever it is the reason at the molecular level and at the cellular level, the adaptability of the cell to avoid the controlling signals develops, and thus the cancer also develops. So as we were discussing, uh, uh, the limitations of the conventional cancer therapy will be achieved with the aid of targeted cancer therapy. So how this targeted cancer therapy is more beneficial to the conventional therapy? It is nothing but it's having higher specificity, and as it is highly specific and it is having improved efficacy, and it overcomes the resistance, and it is highly personalized. So we will be covering this highly personalized treatment opportunity with the targeted therapy when we go and explain the heterogeneity of the cancer. And there are so many drugs that developed for again is that developed based on the targeted cancer therapy, among which 60% cases are highly successful. Still, we are lack of the lagging behind, or we don't have a successful treatment therapeutic regimen for a complete cure of the cancer affected individual. So the reason behind, so as I was mentioning that there are so many molecules that I have developed against the cancer with the targeted therapies, like imatinib is a very golden standard molecule against the BCR-ABL targeting for the chronic myeloid leukemia. Still, there is no permanent cure for the CML. So why so many drugs have been developed, so much is happening, why still there is a lack in the successful therapeutic regimen will be explained by some basic understanding of the cancer heterogeneity. Cancer heterogeneity is one such condition that leads to the development of the failure of the targeted therapy, because cancer heterogeneity develops a different subpopulation in a given tissue or given tumor. So these different subpopulations are characteristics of so many um, abruptly activated signaling pathways, or it is having several clones that having different sensitive adaptability for the treatment regimen. Like some clones will be highly resistant to the current treatment conditions, so they will not be killed upon treatment, and the resistant clone will develop and the cancer reoccurrence will happen. So this cancer heterogeneity is of different types. It is of genetic heterogeneity. Heterogeneity is there, epigenetic heterogeneity is there, cellular heterogeneity is there, and intratumor heterogeneity is there. All these leads to the cancer relapse and the treatment failure. So in such case, how this cancer heterogeneity can be addressed? So this cancer heterogeneity can be addressed by developing different kind of inhibitors, such as dual targeting inhibitors, and multi-targeting inhibitors. So dual targeting inhibitors is nothing but combining two different molecules into a single hybrid molecule, so that they can affect, or they can effectively treat the given cancer. Suppose for a cancer that it is showing heterogeneity for the EGFR and tyrosine kinase region, where um, one single targeting inhibitor that is developed against EGFR will work only on the EGFR, but the other target partner will not be able to treat it. In such case, dual targeting inhibitors are highly efficient to treat such kind of cancers. Another such kind of strategy is multi-targeting inhibitors. The classic example of multi-targeting inhibitors is like cabazitaxel. Cabazitaxel is nothing but a tyrosine kinase inhibitor, but it targets VEGFRs, MET and AXL and other tyrosine kinases also. So thus this employment of this molecule against such kind of heterogeneous cancer develops the improves the overall survival and progression free survival of the affected cancer. The advantages of these multi-targeting inhibitors is nothing but they leads to a synergy effect, and and at very lower doses they will be highly effective and fewer side effects. And onto that, another significant impact of these multi-targeted inhibitors is nothing but with a simple common sense that inhibiting a single target uh, is inhibiting multi-targets is better than inhibiting a single target. So these are all the advantages that are allied with multi-targeting inhibitors. Here is a simple example or some hypothetical model that has developed in a study where they have loaded several small molecule inhibitors to antibody mimicking peptides, and they have prepared a nanostructure device in such a way which is a pH sensitive. So at a different pH levels, it releases a different small molecules, and thus based on the scenario of the tissue and based on the scenario of the tumor microenvironment, based on the scenario of or context of whatever it is, the respective small molecule that is expected to work against a single target will be released and the treatment will be more effective. This is how the more treatment regimens has to be developed for a effective cancer treatment control. So the take home message is that as we have discussed that cancer heterogeneity is having a highly significant effect in the failure of the cancer treatment conditions. As well, the conventional treatments are not very successful enough because of their not because of their non-selectivity. And so to avoid such above things, uh, to avoid the conventional chemotherapy limitations, targeted therapy is fine. However, targeted therapy is being limited because of the heterogeneity and the resistant clone development and all. In such way, we need a novel treatment strategy which comes under the broad term of multi-targeting strategies, where it can be facilitated by combining 2-3 molecules, by doing materials engineering, and by combining effective drug delivery systems. We can it is expected that this approach can be a um, successful therapeutic regimen for the cancer control. So such kind of more advanced things should come arise by having this kind of basic concepts for a successful treatment condition. I take this opportunity to thank for giving this, to thank one and all for your patience listening. Thank you.
Thank you, Dr. Bindu Madhuri, for that valuable lecture. Now, I will hand it over to my co host.
Thank you, Aditya. Now, as we conclude today's scientific session, I would like to invite Dr. Sukant again to deliver the closing talk. Welcome, sir.
I would like to thank all the speakers and the participants. Very happy to see questions, very happy to see very informative lectures. This is part of the broader thing. Our core philosophy is to educate you while giving you career options, while giving you a direction what will get you best work life balance, best earning, best freedom, best research opportunities, best scope to grow. We give you structured things for PhD, for postdoc, for research after MBBS. We give you immigration advice. We give you fellowship advice. We give you advice on how to structure your books, blogs, podcasts, conference organization, teaching, volunteer work, awareness programs, and then collaborate with international researchers. We teach you over 30 AI algorithms, bioinformatics, computational biology to complement your existing skills in network or life sciences, computational life sciences. We make your career go towards drug discovery, pharmacology. We go your career go towards cell biology, modern neuroscience, modern cancer biology, and so on, so forth. If you're interested in not just some degree, but interested in international guidance, which shapes your career, your research, fills up your teaching holes, and provides you overall structured support, look at our 10-point program at aividya.in or ioncurex.com. We welcome people to join us and contribute to the society through teaching, through publications, through products, through apps, through overall development of their own career, and hence in the process, development of science. Thank you very much for being with us. Amazing work, everyone. Thank you. Bye bye.
Thank you, Dr. Sukant. Please wait, we have our next speakers who are two distinguished researchers who bring valuable academic and mentorship insights for students aspiring toward research and PhD, masters in research, alternative career to medicine opportunities. First, we have Dr. Brooks Robinson, Research Program Director and Assistant Research Professor at the Lyda Hill Institute for Human Resilience, UCCS, whose work centers on academic development, resilience, and mentoring students for successful doctoral pathways. And next is Dr. Alfredo Gezzi from the Department of Biology at the University of Puerto Rico. Dr. Gezzi's research focuses on neurobiology, behavior, and the molecular mechanisms underlying addiction and adaptation, while also mentoring students in navigating higher education and research careers. We are honored to have Dr. Alfredo Gezzi before we move into these special mentor talks, I would like to sincerely thank all our student lecturers, mentors, and audience for making today's session so meaningful. Your enthusiasm and presence have made this first day of the lecture series truly valuable. We also look forward to welcoming you to the day 2 of the lecture series on 2nd of May, where we will be continuing this journey with two exciting themes on neuroscience. Foundations of neuroscience followed by mechanisms of neurodegeneration. So these sessions will explore the fundamentals of brain function, neurological disorders, neurodegenerative disease mechanisms, and current therapeutic innovations. With that, we now conclude today's live session and invite you to enjoy these final mentor talks by Dr. Brooks Robinson and Dr. Alfredo Gezzi. Thank you once again, and we look forward to seeing you in the upcoming sessions. Thank you.
What if I told you that getting into your dream PhD program wouldn't come down to your grades or even your research experience, but to how well you play the game? Hi, my name is Brooks Robinson, and I've been on both sides of the table. I've applied to PhD programs myself, but I've also reviewed PhD applications, mentored students, and watched brilliant minds get overlooked because they didn't know the rules of the game. Let's face it, most students don't fail because they're not smart. They fail because no one taught them how the system really works. I'm part of a team of professors, scientists, and experts led by Dr. Sukan Khurana. We've helped students from all over the world get into top PhD programs in Germany, France, the Netherlands, and beyond. We know what works: how to write a letter that lands an interview, how to email professors and get responses, and how to write a CV that helps you stand out from the hundreds of other applicants. This is not guesswork. This is strategy from the other side of the table. Instead of stumbling through the process, learning from your mistakes, learn from ours. We've reviewed hundreds of PhD applications and helped shape even more. Why go it alone when you can be guided by the people who make the decisions? If you're serious about a PhD in Europe, stop guessing. Email us at contact@ioncurerx.com with your CV and goals, and let's build you an application that gets results. Because success isn't just about hard work, it's about smart work with the right team. Join the students who've made it because they have the right mentors. You could be next.
Hello, I'm Alfredo Gezzi, professor and director of a graduate program, and someone who has spent years reviewing PhD applications from around the world. Let me tell you something honestly. Far too many brilliant students don't make it through the door, not because they lack potential, but because of avoidable mistakes in their applications. Here are just a few common issues I see again and again. A generic motivation letter that could apply to any lab, anywhere. A CV that reads like a shopping list, not a story of academic growth. Students who fail to connect their past work to the lab they're applying to, and surprisingly often, poor communication with the potential supervisors. These things may seem small, but trust me, they are deal breakers. But here's the good news. You don't have to figure this out alone. I work closely with Dr. Sukan Purana and an international team of professors and scientists. We have sat on admissions panels. We've guided hundreds of students, and now we're here to guide you. We understand what admissions committees are really looking for, and we are ready to help you present your story in the best possible light, tailored, strategic, and authentic. So, if you're serious about pursuing a PhD, especially in Europe or North America, don't leave it to trial and error. Reach out to us at contact@ioncurerx.com. Let's work together to make your application not just good, but unforgettable.